Thank you for joining me. The main reason I really wanted to speak to you was because we seen that you had done a lot of work and you published review articles recently on sort of mucoadhesion and permeation through mucus. And I thought it'd be quite nice for us to have that conversation. And so, yeah, thank you for joining me.
Yeah, thank you for your interest, and it's a pleasure to be here to speak about these topics.
Excellent. So to begin with, there were a few, I think where we could begin is with mucoadhesion, right? So it's the first sort of few papers that I've seen of yours that really got my attention. Let's start off with the fundamentals and then we can build into sort of the more complex sort of discussions that you've had within your paper. So firstly, what is mucus and why is it a problem or even a challenge for drug delivery?
Okay, so mucus is a secretion that is secreted by special cells, by goblet cells. They secrete mucins on mucosal surfaces, and mucus is like a gel that incorporates this mucins and some other stuff, and it serves many functions such as protection function, lubrication, moisture moderation, and so on.
Okay. And following on from that, so we have mucus in all different sort of parts of the body, right? Well, is it a challenge to drug delivery and why is it a challenge to drug delivery at the moment?
Well, in a way, it's both a challenge and an opportunity because, well, mucus provides some defensive barrier. So it will, from this point of view, is a challenge because in some cases, it prevents penetration of drugs to the epithelial cells. But at the same time, it is the opportunity because mucosal surfaces, they're still in many cases, they are more permeable compared to skin, for example, and this offers an opportunity of achieving better drug bioavailability when we deliver drugs via mucosal routes.
Okay, so it has pros and cons, right?
Absolutely.
Okay. And so I guess we can start heading a bit more in towards sort of what's really been done then. In order to exploit this opportunity that it presents of sort of permeability, I've seen that you have recently done a lot of reviews in terms of looking at that opportunity to improve mucoadhesion to allow certain drugs to sort of permeate through the mucus. What was some of the key findings that you have seen, or what advancements are you seeing in that area of mucoadhesion?
Well, this area originates like from the previous century. The first publications I found is 1947, and these papers were in the dental area. So, like when dental researchers were trying to administer penicillin in the oral cavity. Then, interestingly, that they published it in dental journals. Some American researchers published it in dental journals, and there was like a quite a significant time gap between when these papers were picked up by some other dental researchers, because 1947, it was like a pre-internet era. I remember this time because when I was a young scientist, I remember the time when you need to get information from the library, you need to work with physical copies of the journals, and obviously 1947 is definitely even before my time. So at that time, this paper was published. Subsequently, it was picked up by some other dental researchers. There was another paper, 1949, and then there was a gap until 1980s, where it was sort of picked up or maybe even developed independently by several pharmaceutical researchers. There's some researchers from Japan, Professor Tsuneji Nagai, who is viewed as one of the pioneers in this area. There's also researchers from US like Nicholas Peppas, and from Europe, Robert Gurney, who started publishing in the early 80s on this concept. So then after the 80s, this concept started getting picked up quite quickly by various pharmaceutical researchers. And now if we look at this field, then the mucoadhesion is quoted in many hundreds of papers published each year. So it's becoming really popular. But there were also like important milestones in this field, because initially people have used conventional polymers, conventional water-soluble polymers, because all water-soluble polymers are mucoadhesive to some extent. More recently, there was a proposal to develop so-called second generation mucoadhesives, the polymers and the systems that could form covalent bonds with the mucosal surfaces. And this resulted in like a bit of a revolution in this field, resulted in lots of studies coming from many groups, including my group. And then subsequently, another important milestone is the development of the so-called mucus penetrating systems. And mucus penetrating systems are they act in an opposite way. In a way, they are not mucoadhesive at all, and they are particles coated with non-adhesive short chain water-soluble polymers that do not interact attractively with the mucin components, but because they non-interact with this, they don't interact with the components of the mucus, they provide penetration. And still, well, through the analysis of literature and through some of our experimental data, I can't say which ones are better. Well, it very much depends on the dosage form, on the route of drug administration, and possibly on the individual drug molecules that are used for a particular therapy.
Okay, there's something you mentioned there that's quite interesting that I picked up on. So you mentioned covalent bonding, that is one of the sort of significant points in that development of that journey of mucoadhesion. When you develop a polymer that's sort of covalently bonding, what type of adhesion are you seeing? Because usually when we say covalent bonding, we think of more permanent sort of attachments, right? Whereas if you think of ionic or charge-based, it's very temporary. How does that work then, that covalent sort of system? Does it allow for very prolonged attachment, or is it a temporary? What type of adhesion are you seeing?
Okay, so in terms of how mucoadhesion works, I kind of need to explain a bit more into how the conventional polymers they adhere. There's multiple mechanisms in a way. This will involve some physical interactions such as hydrogen bonding, electrostatic attraction, and so on. Then there's also polymers diffusing into the mucus gel, forming interpenetrating networks. There's involvement of some ions like calcium present within the mucus, and this could provide additional points of bonding. There's lots of theories about mucoadhesion of conventional systems. But then when it comes to the covalently bonding systems, they obviously are capable of forming covalent bonds with mucins. And because mucins are not like mucosal surfaces are very dynamic. So the mucins are continuously produced and they sort of flow on the mucosal surface with different speeds, different lifetime. So if we design a covalently bonding systems that will stick to a particular mucin macromolecule, it doesn't mean that it will stay on the surface permanently. It will still flow. It will still be cleared after some time, but it will extend the duration of contact, which will be of great advantage for some delivery systems.
I see what you mean. So the mucins themselves, they have a lifespan, as you say.
Absolutely.
And so because they're on the surface, they will stay there for a period of time, bond with the polymer, but they will eventually be renewed. And then that is the opportunity for the drug to move into the target site, is essentially what you're saying.
Yeah.
In one of the papers, there was something that stuck out to me was they mentioned the thiol-based polymers. And so I'll put up that table up in this conversation so people can see it as well. It mentions, there's over 450 publications on thiols, and I think it was 29 patents. And they see and it's in commercially available as well in things like eye drops that are used for dry eye disease. Why, and looking at that table, it was sort of the leading polymer type system. What is it about the thiol group that makes it so popular? Why are we not seeing sort of growth in other areas, or are we seeing growth in other areas and it just wasn't included?
Well, thiol groups, their use in mucoadhesion historically comes from the pioneering research of Professor Andreas Berkop-Snurk from University of Innsbruck in Austria. So his group pioneered the use of so-called thiomers, and they introduced this term. And thiomers refer to polymers which were chemically modified by introducing some thiol groups. How thiol or thiomers will work in terms of mucoadhesion? Thiols, they are quite reactive groups, and they are easily reacting with the thiols present in mucins, because in mucins we have cysteine domains, and cysteine as an amino acid, it contains thiols. So thiol groups could form so-called disulfide linkages, SS, when one sulfur comes from the cysteine of the mucin and one sulfur comes from the polymer. So they form under physiological conditions, typically in the presence of some oxidation is basically the reason for formation of disulfide linkages, and on all mucosal surfaces we have presence of air, oxygen, and so on. So that's why thiols are quite reactive and polymers modified with thiols are quite adhesive. There's also a bit of a negative aspect of thiols as well, because thiols could be oxidized in air, which
Yeah.
means that this could lead to some stability issues if we have these excipients, especially if they are in solutions, in aqueous solutions, then they could potentially be oxidized upon storage. In solid state and dry state, they are a bit more stable,
Okay.
which did result in subsequent studies from the same group in Austria. They did develop protected thiols in their system. So polymers with thiols, thiol groups are chemically protected, and then they could be deprotected in situ, in vivo, and could stick. But then when I came to this field, I visited Professor Berkop-Snurk laboratory in 2006. I've been there twice in 2006. And I've learned about these technologies, and I kind of started thinking about where I could contribute, because it was already an established field. And as a chemist, I'm chemist by my background, I started thinking about some other chemistries. And then I came across a couple of papers from Technion in Israel. There was another professor there called Havazelet Bianco Peled, and her research group pioneered the use of so-called acrylated polymers. So the principle of acrylation is, again, they were taking any water-soluble polymer. They demonstrated it in a couple of examples. They took a water-soluble polymer. They introduced acryloyl groups into these polymers. And acryloyl groups, they also have a high reactivity towards thiols present on the mucins, but the chemistry is different. This chemistry is called Michael addition reactions, also called thiolene chemistry, thiolene click chemistry. So I was really inspired by this research, and at some point I've realized that, I need to continue this field and to expand the number of systems. And in my group, we started looking at various systems, various polymers with conjugated unsaturated groups. So we introduced the maleimide groups, for example. Then we demonstrated that polymers modified with metacrylyl groups could also enhance adhesion. Later on, we expanded this to some crotonyl groups, to itaconic acid groups, and so on. Additionally, if we look at the literature, then there's some groups, they use completely different principle of binding the polymers to mucosal surfaces. There's lots of studies about the phenyl boronic acid conjugated to polymers, and phenyl boronic acid could form covalent bonds with the OH groups present in mucins. We also have done a bit of work in this area. Also, we worked on introducing aldehyde groups, and aldehyde groups could bind to amino groups in mucins, and this binding is due to Schiff base formation. So there's a range of chemistries available at the moment.
You've spoken about a lot of different groups and interactions with mucins. Where are you seeing the most sort of promising results of mucoadhesion?
Well, at the moment, what is in this area, as you mentioned previously, this table, where there's lots of thiolated systems available both in academic literature. There's few already hit the market, and again, it is the development by Professor Berkop Snurk. One of his thiolated polymers, thiolated chitosan, is now on the market as Lacrimera, as ocular lubricant to treat dry eye syndrome. There's no, as far as I'm aware, there's no other examples of other chemistries that have hit the market. But well, who knows? Maybe the research and development will continue, will progress, and hopefully some of the systems we introduced may also reach the market. Because they may offer some advantages. For example, the systems with unsaturated groups, they are not unstable upon oxidation. They show better stability.
Okay. There's something that I wanted to really speak to you about was this sort of the clinical application of a lot of these polymers. And because when we talk about mucoadhesion, as we previously said, you've got a lot of different sort of areas that we can say are sort of mucosal surfaces right throughout the body. And depending on sort of your target, you will want different durations of mucoadhesion. Is there any systems that you know or any standardized way of testing mucoadhesion? So for example, if it's nasal or buccal or whatever your target is there one sort of protocol for each type of organ that you would be aiming for?
Yes, it's a very good question, because in fact, there's no standardized systems, there's no standardized methods. Different researchers and even different companies in pharmaceutical industry, they use different approaches. So some researchers, they use semi-artificial mucins, so basically mucins that were isolated from pig stomachs, and they are commercially available. But these mucins, they are not like, because of their purification, because of their storage, they are no longer the native mucins. So many people use these mucins because they're easy to get hold of, they are of relatively stable properties. And that's why people use them and demonstrate that, yes, this polymer interacts with the mucin. A bit more real scenario would be to isolate mucins from the like fresh animal tissue. But then again, the methods involving mucins will be the methods proving that yes or no, yes, there's an interaction between this polymer or between this system and the mucin, or this interaction is absent. These methods are useful because they help to understand mucoadhesion, but I strongly believe that if the real mucoadhesion should be tested, it should involve the use of real animal tissues, which creates some ethical dilemmas because, well, especially in the early research, many researchers, they've used tissues taken after killing animals, after killing laboratory animals. You could trace all these studies in the literature that people mentioned that they sacrificed rabbits or rats to generate tissues, and then they've used for adhesion. In my group, we use the tissues from the local abattoir, so we are not sort of ethically constrained from this point of view. And we also use techniques that both available in the literature, and we developed some of our own protocols. One of the very common protocols that was developed many, many years ago is so-called tensile test. When you use a mechanical machine, you attach your dosage form to a probe of the mechanical machine. Then on the other end, you will have ex vivo animal tissue, mucosal tissue. You put them in contact, and then you apply force to withdraw your probe, and you measure the forces required to detach a dosage form from an animal tissue. Then there's special instrumentation available for that purpose. For example, texture analyzer. It could record the detachment curve. Then from this curve, you could calculate the detachment parameters. You could calculate the work of adhesion. That's one of the more or less standards in the field. But again, the standard has limitations because, well, in the natural environment, when we apply a dosage form, we don't apply these forces. So typically the dosage form will be washed off of the surface. Therefore, there's researchers who developed the so-called flow-through techniques. And we, in my group, we've optimized and we further advanced this flow-through technique by introducing the fluorescence techniques. So we developed at Reading, we developed the fluorescence flow-through assay when we use a fluorescently labeled formulation. We put it on the animal mucosal tissue. We wash it with the biological simulated fluid. For example, if we work with ocular drug delivery, we wash it with simulated tear fluid. If we are investigating something intranasal drug delivery, we use simulated nasal fluid. And then we take images. We take images using a fluorescence microscope, and we see how well the fluorescence disappears from this surface. And nowadays, there's free software called ImageJ that could be used to process each image and to convert the image into the numerical information. This way we could generate the information that could be analyzed using statistical methods. We could sort of mimic the environment where there's a continuous flow of a fluid. But even this method has limitations because you can't use the flow at the same rate as it happens in the human body. So there's lots of methods potentially. There's lots of other methods. I could possibly talk for hours about these methods.
No, I think that's really helpful that you've mentioned that because, like, for me, it's not anything I've not worked with mucoadhesion before, so it's quite a new area. You know, this conversation for me is quite new. So thank you for actually explaining that because so I like the fact that you guys have sort of moved to a fluorescent model, the fluorescent flow through, because when you've got sort of the conventional methods that you talk about of, you know, sticking it to a probe and measuring that retraction force, it's you can measure the adhesion, but then that adhesion doesn't necessarily replicate what's happening in vivo. So you do, you know, you get values and you get an understanding of that attraction, but not necessarily in that diverse sort of in vivo area where you've got saliva or mucus also having an impact. And one of the things, so I did a bit of work with ocular as well, and the issue was, you know, you can measure things as in how well they stick to the tissue, but then you've also got the issue of the other organs in the area. So when you've got your eyelids blinking as well, that adds such a complicated layer to sort of recreate ex vivo, you know, in the lab because the tear flow rate is actually very, very low. But then because you're blinking, it just spreads everything, and then you've got mechanical forces. One of the things you previously mentioned is, and correct me if I'm wrong, but you mentioned that the properties in the aiding sort of polymer, for example, they're very different when you're trying to achieve permeation compared to mucoadhesion. Can you explain a bit more about that? What do you mean by properties, but also why are they different? What would you look for in, let's say, permeation enhancement that you wouldn't look for in mucoadhesion?
So permeation enhancement is, in a way, is a very different concept.
Okay.
There's like the whole field of researchers who work with so-called permeation enhancers. Mostly the permeation enhancers, they are used in transdermal drug delivery because our skin could tolerate some more active chemicals that could help the skin getting more permeable towards the drug molecules. In mucosal areas, there's also some developments in permeation enhancers, and we've done some work in the ocular permeation enhancers. At the time, and again, it was more than 10 years ago, we were interested in an ocular condition that is called keratoconus. And keratoconus is a condition when the cornea of the eye is thinning and becoming like less mechanically strong, so and is getting deformed under the effect of intraocular pressure. So one of the very unusual way that clinicians use to treat this condition is actually involving the physical removal of the corneal epithelium with like a spatula under local anesthetic. So basically they scratch off the top layer of the eye. They do it because this epithelium is highly impermeable. So once they remove this epithelium, they apply the drug called riboflavin, and they apply it for like half an hour instillations of riboflavin in order to saturate the rest of the cornea with riboflavin. And why riboflavin is needed there? Because subsequently they use UV treatment with UV light, and riboflavin facilitates the cross-linking of the collagen present in the cornea. So basically, once this operation is done, then the patient is discharged, and the cornea is more cross-linked, so it is mechanically stronger. But obviously, removing the epithelial layer of the patient, it is at least uncomfortable, but then there's the post-operation consequences. It is pain, it is prone to infections and so on. And at the time when I talked to one of the ophthalmologists who told me about this condition, I became interested in permeability enhancements to avoid this epithelial scrape off. And then we started looking at different permeability enhancers and identified the most common ones like cyclodextrins, some chelating agents, and we started looking at ex vivo experiments formulating riboflavin with these enhancers, looking at how enhancers could facilitate permeation through the cornea without the removal of the epithelium. All these experiments were done with ex vivo tissues. And we've identified even a new class of enhancers that has never been reported. We've basically demonstrated what is the mechanism of enhancement. In some cases, it was like the enhancer was extracting the cholesterol from the epithelium, making the epithelium more leaky. In some other enhancers, for example, chelating agents, they extract calcium ions in the tight junctions between the cells there. And well, another sort of error, another failed story that we, in all these experiments, and it was like a PhD student. I had a very good PhD student who was doing these experiments, but we've used cow's eyes at the time, and then the whole PhD was using cow's eyes, which we were getting from the local abattoir. But then only a few years after, I've realized that cow's eyes are not the greatest model because they have much thicker cornea, much thicker epithelium. And I wish I had another chance in a way that if I will have maybe another student, another opportunity to look at it again, then I would definitely use pig's eyes, which are anatomically, physiologically a bit closer to humans.
Okay. Yeah, I wasn't aware that they were a bit thicker, the cow's eyes. I didn't know that.
Yeah, in fact, in subsequent research with another PhD student, we did look at comparison of different anatomical, physiological features of different eyes from different animals. We haven't completed this research yet, and it's not even published, but then I wish I could get more researchers to continue this, because in this case, we would look at specifically permeability of drugs through different eye models and things like that.
Yeah, I mean, once you do finish that work, do let me know, because that's something I would love to review. As in,
thank you.
I think that's very important. The ocular space is very unique, and I think that corneal barrier has been an issue since since the beginning of sort of the trying to treat any corneal disease, because like you mentioned, it's so impermeable, but then researchers, we don't really have good model or even understanding of different models yet, I don't believe, in terms of what's the closest to the human eye. So once you do have that, let me know, because we could even probably have another conversation like this where we break down sort of the science behind it.
Absolutely.
Has your work been published, the one that you mentioned in terms of looking at the permeation of riboflavin with the cyclodextrins? Is that
Yes, we did publish a series of studies. Uh, we did not achieve like clinically relevant, clinically achievable results, I would say. And again, I possibly should blame the cow's eyes. That's why I really want to come back. But then we did establish some new classes of enhancers, and for the known classes of enhancers, we've actually established the mechanisms of enhancement, which was important. Actually, these papers do receive lots of citations, so I guess they sort of I hope they inspired someone else to do it. I haven't followed the keratoconus research in the recent few years, but from time to time I see that these papers appeared and new papers appeared, and then they do cite our research. So I guess it was not a waste.
Yeah. And so you mentioned that you use cyclodextrin as one of the enhancers.
Yep.
Was that the only enhancer you used or looked at, or were there others as well?
No, we did use cyclodextrins. We did use chelating agents like EDTA. We did look at some EDTA relatives of having the same structures, and molecules like EDTA, they bind calcium, and they could extract calcium from the tissue. And the new class of enhancers we introduced is called crown ethers. And crown ethers, well, they are really reported in the pharmaceutical applications, but they are very interesting molecules. But more recently, we've done some studies with crown ethers, and we found that there's a bit of toxicity there. So they have some toxicity. We still haven't published the toxicological evaluation of crown ethers, but possibly it's been discovered even before us that crown ethers are toxic. But crown ethers do provide some unique enhancement properties for certain drugs.
Yeah. Out of the sort of enhancers that you use, so your cyclodextrin, crown ethers, the chelating agents, which you mentioned that they weren't clinically up to the level of concentration you needed, but where did you see the best sort of permeation enhancement amongst all of your enhancers that you used?
I don't remember exactly which class did enhance it better. They kind of were providing some comparable enhancement potential. We even did try some weird stuff, weird compounds as enhancers, and we did identify some very interesting trends. But unfortunately, again, is the case that I need more people to continue working on that.
No, that's fine. If it's published, I can find it anyway, the data on those, and I could put it up in the video. The crown ether was something that you mentioned in the review article as well. So in your ocular, I think there's a review article that you published that looks at just ocular permeation, and it's quite interesting because you mentioned the mechanism of a lot of these enhancers of sort of cyclodextrins, crown ethers, EDTA, in terms of calcium removal. The crown ethers was very interesting because it's something I hadn't read before. And what is the mechanism by which they sort of cause enhancement of corneal permeation?
So for corneal permeation, what we've established that crown ethers, they also bind calcium. Crown ethers are very unique molecules. They're like a cyclical analogs of PEG, but obviously much smaller molecular weight PEG. And they have a unique capacity of binding ions like alkali metal ions and alkali earth metals like calcium, potassium, sodium, and these sort of things. In fact, if I'm not wrong, there was a Nobel Prize awarded for the crown ethers for their binding ability of these metal ions. And there's some technical applications for crown ethers, but I haven't seen many pharmaceutical applications. But what we've established, we did experiments when we did use cow's eyes. We treated them with crown ethers, and we did look at calcium extraction, and so we basically have proven that the enhancement in the drug permeability is related to the extraction of calcium ions from the cornea.
Okay. And what is that relation? What is that relationship then between calcium extraction and permeation? Why are the two linked or associated?
What I believe and what I know of are that calcium ions are present in the cell tight junctions. So if these ions are extracted by any compounds, then the tight junctions become leakier, so they become more permeable. That's why it results in permeability enhancement.
Okay. The other thing sort of I read in that review article was that crown ethers have hydrophobic and hydrophilic domains, which can help sort of drag the drug through the different hydrophobicities of the cornea as well, because you've got the sort of tear film and the oily and the watery layers. I think it was something along those lines that you mentioned as well.
Yes, crown ethers, they do have some hydrophilic groups like oxygen and some hydrophobic groups like CH2 groups. And then because they are not like planar in their structure, so they form like they have hydrophilic surface and a hydrophobic surface. And I guess it could help permeability as well. But this will require more research to be done in
Yeah.
terms of to understand what happens with the actual crown ether molecules, whether they do permeate through the tissue or not. It will be a bit more challenging to establish that because
Yeah.
crown ethers are difficult to detect within a biological tissue without any labeling. But then if you do fluorescent labeling, you're essentially converting a crown ether small molecule into completely different molecule.
Okay. I see. Yeah, no, that just caught my attention because I remember seeing in the review distinctly there was two statements. One was about the calcium extraction, the other one was about its sort of amphiphilic nature.
Yep.
One of the things that I seen as well that really stood out to me throughout the review was, well, it was in the other paper, not the ocular permeation, but there's another paper you published in terms of a review article for mucus permeation. There's a lot of talk about sort of the PEG molecule helping to move through the mucus. And it's pretty much seen as sort of a gold standard. Have other polymers come about that have rivaled PEG that you know of, or is PEG still gold standard?
Yeah, it is a relatively new concept, basically to develop so-called mucus penetrating particles or mucus permeating particles. So if you have, well, let's say if you have nanoparticles loaded with a drug and you coat them with low molecular weight PEG, typically like a PEG of 500 or 5,000 Daltons, then this will create so-called stealthy surface, and that will help the particles moving through the mucus. When I came across this study, and this study was published by Professor Justin Hayes from United States, I was really inspired with his publications. And I started thinking, well, because I'm polymer scientist originally, so a polymer scientist in me started thinking, oh, what else could be used? And at the time, I also was researching into the polymers that are called polyoxazolines. And I've realized that there's a potential there. So we found some commercially available polyoxazolines, low molecular weight, that could have like reactive groups. And we designed model particles that could be decorated with polyoxazolines. We also fluorescently labeled these particles and subsequently used them in diffusion studies. So what we have established that particles decorated with polyoxazolines, they could diffuse at sort of the same levels as the pegylated particles.
Okay.
In a subsequent study, we published this paper, but then because of that paper attracted interest from collaborators, and one of my collaborators is Professor Richard Hugenboom from University of Ghent. He's one of the really big scientist in the area of polyoxazolines, and he's synthetic polymer chemist. So his group has synthesized different polyoxazolines for us, and these polyoxazolines were different in their like hydrophobic hydrophilic balance. Then we tried their polymers, again, attached them to the surface of our particles, looked at the diffusivity, and we found that like methyl oxazoline, methyl polyoxazoline is the best, is in a way the most hydrophilic out of the polymers we've used. Subsequently, we have done some work on again with the Hugenboom group again, when we looked at some other polymers, and in a subsequent study we published a paper on chitosan with grafted non-ionic polymers, including PVP. PVP is polyvinyl pyrrolidone, is one of the most common pharmaceutical polymer. So if you modify the surface of particles with short chain PVP, you actually could achieve the same permeability enhancement effect. So yeah, there's lots of potentially there's lots of polymers that could be used. There's still some researchers that still continue doing this research. I also wish to continue it. Again, I need more people to do it, but I have some more ideas that I wish to explore in this field.
Yeah, the polyoxazoline work was very interesting, I think, because when you look at the different chain lengths, I think it was methyl, ethyl, and then propyl.
Profile, yeah.
Yeah, you can clearly see the difference in diffusivity with changing that, shortening the chain length or increasing the chain length, because the more hydrophobic you get, the sort of reduced diffusivity you get. So I'll pop that on the screen for people to see as well. The graph was quite nice. I think it was a very nice piece of work. When we're talking about sort of, we mentioned that mucoadhesion and mucopermeation, they're very different sort of concepts, right? They're very different fields. Is it possible to have a polymer that can do both? So I know we've previously said that, for mucoadhesion, you can optimize this, or permeation, you can change sort of the chain length and things like that. But is it possible, or do we see polymers like this currently in literature that do both, that can be utilized?
Potentially, it is possible. I even had a discussion with like a synthetic polymer chemistry colleague very recently about this concept. Potentially, you could create particles that have mucus penetrating properties. Let's say they're pegylated, but then once they penetrate into the layer of mucus, the peg starts to come off if you create like cleavable bonds, and then the mucoadhesive component is released, or mucoadhesive surface is released this way, and then it will adhere. So potentially it is possible. We've never done it yet, but very recently I had a discussion with someone about it.
Sounds like you would need a very dynamic system, something that
Yeah,
can move with the environment, that's when I think things get very interesting, when you introduce the
absolutely.
dynamic. Okay. So from my perspective, I think, the main reason I wanted to sort of have as I mentioned at the beginning of the conversation, was because of your background in adhesion and permeation was quite interesting. And you've answered pretty much all the questions that I've had in mind. Is there anything we've not covered, or something that you would want to bring up?
Well, with regards to mucoadhesion, I guess now I started moving into a slightly different area. That's like the area that involves some alternative testing methods. And I became interested in life invertebrates as models. And these life invertebrates, they could be used for testing systems for transmucosal drug delivery. And we've established a new laboratory full of different life invertebrates. For example, we've done some research on the worms called planaria. We've developed a new method of using planaria to predict irritancy properties of the dosage forms and of chemicals. Subsequently, we've also used planaria for testing some of the drug delivery perspectives or possibilities. For example, we looked at psychoactive drugs, how they could affect the worms. So that's what makes me really busy in the last few years. But then I also link this invertebrate models to mucosal applications. Well, when it comes to planaria, obviously we developed the mucosal irritancy models to predict the irritancy of our formulations. We also try to establish some possibilities of testing some of the mucosal dosage forms for their efficiency to deliver antibiotics. So we use the infection model. So that's what I wanted to mention as well.
It's been a pleasure, and thank you for joining me.
Okay, thank you very much, and it's been a pleasure for me as well.